Hello, everybody. So I'm presenting today Randall Wisser, who is a research director working at the Department of Plant Biology and Breeding of INRAE in Montpellier, France. Let's say he's also an affiliate faculty member at the University of Delaware. He's coming from Delaware yesterday. Dr. Wisser works on population adaptation to the environment at the convergence of quantitative genetics and plant ecophysiology. Today, we are very lucky to have him in person for this international plant breeding seminar series. Please join me in welcoming Dr. Wisser.
Thank you very much. Happy to get started quickly here. I've got a few areas I'm going to try to cover at the time we have, so hopefully it's not too overwhelming. But thanks for the introduction, and thank you already, the center for supporting this visit. And I've already had some great, interactions, meetings with folks on that. That will continue today, and I appreciate the time that I have with you all here. So first, I just quickly, for those of you who don't know, I want to just rapidly share with you about where we're at. We're in France here, located near the Mediterranean Sea, in the city of Montpellier, which includes very close proximity to a lively city, mountains, and the sea itself. The city is about 300,000 people, with a metropolitan of about 800,000 people in the area. But important for this group is just a very beautiful place to live, sunny most of the time, and really enjoying ourselves there. So, but important for you all is to know, for those of you who might not know, Montpellier is a major international hub for agricultural research, with a large number of researchers in agri-food and environment research. There's about 30 research units across different institutes. This includes INRAE as well as CNRS, 2 national institutes, 2 university structures, particularly the Institut Agro, which is the school where we're located within our unit, but as well as the University of Montpellier. In addition to international institutes such as IRD, International Research and Development, CIRAD, and CGIAR headquarters is located in Montpellier. So it's this very, this place with many activities in the space of, ranging from plant science out to ecology and other areas of agricultural research. I'm at the LEPS unit, the Laboratory of Ecophysiology of Plants under Environmental Stress, and we have 3 teams in the unit. The teams include MAJ, which is the team I'm within, called Modeling and Analysis of G/E. And then ETAP and the MAJ team is focused primarily in cereals, specifically wheat and maize. And ETAP is a grapevine group focused on dry environment adaptation. And M3P is an engineering and phenotyping team that has developed phenotyping infrastructure and development of computer vision techniques for phenotyping technology that I'll share a little bit with you today. So what I'm going to cover today is how the work we're doing is combining phenomics, genomics, and modeling. Charlotte mentioned how we're focused on this approach to try to integrate quantitative genetics and ecophysiology that builds off some of the history also that has happened at LEPS before my joining them. And I want to briefly, this is kind of a little quick outline of what I'm going to talk about. I'm going to talk about population adaptation as a meta process. And then I'm going to focus on flowering time, which is what we're using as our kind of proof of concept to study some various topics about adaptation. And I'll briefly highlight stories about mechanisms for G/E. So these will be like little vignettes with some findings and the directions and the places we're going. Talk about mechanisms for G/E, experimental evolution in the context of like breeding populations that we've experimentally evolved, geographical adaptation, and model-based coupling of genetic and ecophysiological models. So the way that we're starting to think about adaptation is not as basically a 3-way interaction. We're trying to think more comprehensively, let's say, about adaptation, that it involves this interaction between genetics, the building blocks of heritable variation, of course, plasticity, that is the phenotypic modifications that occur throughout the lifespan of an individual, and evolution, the forces of change, such as population genetic type shifts in the population. So the main point being that we want to think about this in a more systems level way and orient ourselves towards trying to model that to understand how populations adapt to different environments. And the framework for that builds from all of the amazing work that our communities have advanced. What this is kind of highlighting are several just, images showing how the field of ecophysiology and much research has advanced our understanding of developmental plasticity, how plants respond to different environmental factors, creating reaction norms to specific environmental conditions throughout their growth cycle. And of course, we all know here in this group, we're talking to the way that quantitative genetics and genetics has advanced in a number of avenues, understanding genetic variation and characterizing genetic variation with prediction models, and et cetera. But now we're in this space, we're talking more about genetic variation as to the within individual plasticity that happens across different environmental conditions. And then how those interact in the context of evolution or change. So you can even think how G by E itself is not this static thing in a population, but it's changing with changing with the context in which we shift our populations over time. So there's several studies that are highlighted here that kind of cover these different areas. So we're focused on flowering time. We've been focused on this for quite a while. And the major reason we have remained interested in this is because of its central role of flowering time in both the geographical spread of crops and their crop productivity. Shown here on the left, you have the fact that maize's origin in Mexico has spread. Maize has spread throughout the world and has achieved that spread in part by its ability of its flowering time to be manipulated for geographical adaptation. And secondarily, crop productivity. We've been working towards, the reproductive transition of flowering time. It defines how the canopy structure gets laid out. And we have a postdoc in our group who's been focusing on trying to understand how to compare diversity in terms of the way in which the canopy forms as a function of floral transition, the timing of the floral transition during the life cycle. And what these graphs show here is how the size of the leaves, once aligned by the timing of floral initiation for diverse genotypes of maize, start to show you how the canopy structure is highly normalized. It follows a process of coordination rules in the development of the leaves until the time of floral initiation, followed by a successive cycles of development where the leaves change their patterning across time. And that connects both with the sheaths and the internodes. So another reason why we're heavily focused on flowering time at this stage. Of course, there's many people who have been involved in a lot of the work I'm going to talk to you today about, but current people that have been in the group or have graduated from the group in the time that since I arrived at INRAE include the folks mentioned here. Today, I'm going to talk a lot about some of the work from Justine Drouot, who was a PhD student with us that just completed her work on characterizing ecophysiological variation to dissect adaptation. And I'll highlight the work of Arnaud Desbié-Piat, who worked on integrating, virtualizing, connecting genetic variation with crop modeling to virtualize adaptation. And the person carrying on that work now is Lorenzo Rocchetti, who's a new postdoc in the group. And Christian Johnson is the one who's been working on this coordination work with respect to leaf canopy development. I'm not going to be able to have time. I'm not going to have time to touch on the work of Emily Pied, who was a postdoc with us, now a permanent researcher at INRAE, and Jonas Rodriguez, who's now a data scientist at Syngenta, but their work helped advance some other methodological approaches to, let's say, deciphering adaptation. We can, for those of you that I meet with, we can talk more about that separately. So the first thing I'm going to discuss is a bit about mechanisms of G by E. And of course, this room knows really well this way that we can think about G by E as separate components. We have scale change G by E and rank change G by E. And the question might be how we think about how these different forms of G by E arise. Of course, we often treat this in a statistical framework in our quantitative genetics community. And what we're trying to do here is provide some mechanistic insight into how this types of G by E form, because these determine how we might think about structuring breeding programs, or how we might implement different selection schemes. So in terms of flowering time, the most important thing to understand about how this G by E can form is this kind of framework that I'm laying out here. This our understanding that the environmental sensing across the crop cycle occurs at different timescales during development. So what I have portrayed here is the crop cycle from planting to flowering, with the floral initiation indicated by these black triangles. And for the plant, temperature is sensed by the plant throughout its entire growth cycle. In contrast, photoperiod is something that is sensed in a specific window of time that precedes the floral initiation stage, or basic floral initiation, I should say. And so there's these two different timescales operating that control the way in which the development timing happens in actual days. That's controlled, affected by temperature and photoperiod. And then the response, the way in which sort of leaves, you can think about, any grass, you have leaves emitting at a certain pace across days or time. And those responses follow different reaction norms. So in terms of the temperature component of response, you have a function that looks something like this. And I draw a blue and a red curves to show you how different genotypes exhibit different rates of development in response to common temperatures. And similarly here, when we look at photoperiod response, we have, in the case of maize, which is a short day species, you have environments where photoperiod plays very little role in altering flowering time. And then there's a threshold at which that is genotype specific and can follow different patterns of response on development after you pass this threshold. And the consequence of that is you have these different reaction norms that are interacting with each other within an environment as the plant is sensing its local environment condition. So to play this out a little bit further, to give you some idea how this relates to scale and rank change G by E, let's imagine those the blue and the red genotypes shown here that undergo they're genetically programmed to undergo floral initiation at a different time. And consequently, in a given environment where you may have a warm temperature condition, the timing of flowering between these two genotypes exhibits some differentiation in days that is separated as portrayed here. And if you look down at the next graph below, you have a cool environment scenario. And in this environment, this gets kind of spread out in time, in days. And so temperature is either slowing or speeding the timing of flowering and the timing of the reproductive transition, and consequently the timing of flowering, in ways that can alter the genetic variance at the scale change level. So temperature is modulating this pace of development. Interactions between genetic variation in development rates with fluctuating temperatures within an environment can create scale change G by E. And this partly explains differences in heritability. If you have an environment that's warm, you have a compression of that variance, and consequently, in days, many more genotypes in the population will flower almost on the same day. You can't differentiate, you don't have as much distinguishing power to see those genetic differences within that environment. Whereas in a cool environment, you have a spreading out of those genetic effects, and you have a better distinguishing capacity. But in reality, you have this other level of interaction with photoperiod. And in the case of photoperiod, what happens is the genotype by temperature interactions also determine the day when a genotype becomes sensitized to photoperiod. As I mentioned at the beginning, the sensitized stage happens just preceding this floral initiation stage. So in a given environment where the temperature is fluctuating in one way, you can of course think across environments. It's not a problem. You go across one environment to another, and you can easily conceive how genotypes would sense a different photoperiod in those different environments. But here what I'm trying to show you is how within the same environment, you can also perceive different photoperiods. And when you look across these environmental scenarios, this is what starts to lead to rank change G by E. So genotypes can sense different photoperiods, and genetic variation in photoperiod sensitivity can lead to these rank changes. And then there's additional scale change G by E that's formed because of their timing of floral initiation being different and temperature playing a continuous role for the rest of the cycle until flowering itself occurs. So we did a simulation analysis that's kind of parameterized from real genotypes and looked across US and European environments to try to and show that within a diversity panel that you might grow any one place, genotypes can experience as much as 1 to 2 hours long differences in day length. There's not sensing the same day length, even though they're in the same place. And so what this first graph here is showing you is how that kind of can be understood. So you have some genotypes that are early per se. They would normally flower earlier in an environment where photoperiod is not affecting their flowering time at all. And some genotypes that are late per se. So their temperature determined timing to floral initiation makes them early or late. And this is the photoperiod fluctuation of a given environment. This would be the planting date, this green triangle. And the orange triangle here represents what an early per se genotype, when it would get to this sensitized stage. And depending on temperature conditions, the late per se genotype could reach that stage at this time on a different day, closer to the yellow or further away from the yellow. And consequently, depending on that interaction between temperature, developmental programming in flowering time, and photoperiod fluctuation, where you're at in the environment, turns out that genotypes can experience different photoperiodic conditions. And what this map shows is just that you tend to see this, of course, more exaggerated in European environments because you have a higher rapid rate of change in photoperiod across days. And therefore, if you're growing a diversity panel of lines, some lines are experiencing a day length that may be 15.5 hours, and other ones may be experiencing a day length of 17 or so hours, even though they're growing them in the same environment. And then let's not forget that those genetic responses to that condition have variation, and it leads to a more complex G by E, even though we're just talking about two kind of simple components of the flowering time response. This graph is just showing the trends as it relates to the rate of change of day length and just how much differences can occur between genotypes that belong to this early class versus if we were to assume that when we model G by E, assuming that in this place, every genotype experienced the same photoperiod, what's the magnitude of difference of making that assumption and how that changes across different conditions. And you can get as much as a 50-minute change relative to what you might consider the reference level for all of your genotypes. So what I just shared quickly was, and hopefully maybe it wasn't the best delivery of making that clear, but I hope that you understand from what I've shared is that the scale change G by E that you see is partly in the case of flowering time is driven, has a strong thermal effect that drives that. And in the case of rank change G by E, this arises from photoperiod. So we start to understand the mechanisms that are controlling those G by E components. So I'm going to switch to experimental evolution. We've been working on a number of different populations that have been experimentally evolved within different environments for early flowering time. In particular, this work all comes out of activity where we're trying to deal with the fact that if you take tropical maize and grow it in the temperate zone, it has extremely late flowering. Part of that is due because of photoperiod sensitivity. And so we did selection experiments for early flowering and characterized the genetic basis of their response and a number of other facets that I'll share a slide about in a second. One of the more sort of ambitious things that we did in a large collaboration with a number of colleagues was a parallel selection experiment where we constructed our own synthetic population from a collection of specific tropical lines. So these are showing like the different tropical lines that were, cartooned here. We recombine them systematically like you would a magic type population. It's actually just synthetic. We didn't inbreed them. And then that synthetic population was distributed across 8 locations representing different latitudes spanning Wisconsin to Puerto Rico. And in each of those locations, we performed selection following the same selection regime, using protocols that were designed specifically to give us strong power of dissecting and characterizing the genetic response. So we basically had very large effective population, or we kept the population sizes large, 10,000 plants in each round of selection, and selected the earliest 500 plants to minimize the effects of drift and response. And this led to a collection of about 20 populations that we registered in the Journal of Plant Registration. So it's a resource that other people can use as an experimental system to study an adaptation, or potentially as a source of diversity for temperate zone breeding programs. So connected to what I just shared you about the G/E, what we observe in characterizing this particular parallel selection experiment population, one of the things that we did with it after the selection itself is we did a common garden multi-environment trial where we grew all 20 populations in all of the original 8 locations for multiple years and measured multiple traits, including flowering time. And what this sort of matrix here is showing you is how when we have an interaction of the selection location against the evaluation location. So in the evaluation location of, say, Iowa here, we had measured the response to selection, shown in this color graph, with respect to flowering time, the amount of phenotypic change for flowering time, for the individual original selection locations. And so what you can see from this graph is you get a pretty marked difference in the level of phenotypic change on average in this kind of upper block. And we know that, of course, is because of photoperiod, that this is about where the threshold is for photoperiod sensitivity. So photoperiod sensitivity, when it's expressed, it causes a longer delay in flowering, and selection will manipulate this component of flowering time. Whereas at southern locations that are under short day environments, the populations just don't respond as strongly because they're really driven only by this well, not entirely only, but primarily only by this thermal component, where photoperiod, the expression of the photoperiod alleles is not expressed, and it doesn't impact the flowering time. We detected signatures of local adaptation. So if we look across the diagonal, on average, there's a higher level of phenotypic change than the off-diagonal averages. This is reported in the study on this, but we basically see this kind of context dependency showing up at the phenotypic scale. We also characterize that at the ecophysiological scale and genetic scale, and see this across the all these different scales. So what this graphs here are showing you is when we, from this trial that we did of the common garden of all the selection populations, we're able to, for each population, separate out the effect of the thermal component from the photoperiod component. And what this shows here is how, going from generation 0 to 2 generations, how the thermal component of flowering time has shifted, has gotten shorter and shorter. So there's just less thermal time required for flowering as selection cycles increased. And that occurred in both the environments that were below the block diagonal, that is actually here, sorry, and above the block diagonal, that is here. So all environments, no matter where we're at. So it's really like a per se effect. Temperatures influencing the population, no matter where you're at, and selection is interacting with that to decrease the thermal time requirements to flowering. However, for photoperiod, we only see a response in that component when populations are selected in the long day environments, which is exactly kind of what we expect. And in short day environments, it looks like it just kind of drifts, which tells us also that there's probably no ecophysiological or linkage effects that are So by even though this trait is shifting and that both temperature and photoperiod affect flowering time, we see really no impact on the photoperiod on the populations where thermal time is showing a change. So that suggests some kind of decoupling of those components of flowering time. Then when we look at the genetic scale, where we've estimated the effects of alleles across the set of markers that we had scored in these some of the populations, what we can see are signatures of both broad adaptation, which is probably the thermal component, whereby allele effects show, a correlated effect, a correlation in their effects between one environment and another environment, whereas other sets of alleles look to be more like conditional effects. They only show positive effects. So sorry, these numbers are probably real small, but the allele effect sizes here are only positive in the environment B in this case, and they're 0 in environment A. So the axis is 0 here and 0 here, and so these would be signatures of local adaptation that are very likely connected to the photoperiodic component. So now I'm going to switch to presenting a little bit about geographical adaptation. I just have a couple slides about this that stem from this related work about trying to think about the G by E, interpret it in the context of experimental breeding, and then now we go back, take a different kind of lens, a broader lens, and examine this from the point of view of geographical adaptation. So in maize, what we did is we leveraged an existing diversity panel that's very well characterized. It's 282 diversity panel, and in that panel is reflective a bit of some of the historical adaptation of maize. It includes tropical material and temperate material, and historically tropical material has come up through into the temperate zone and had underwent historical selection for adaptation, partly flowering time. People have shown how photoperiod was a target of selection in that transition. And what we did is we characterized the two components I just mentioned, the thermal component, like the per se component, and the photoperiod sensitivity in that panel. And what you can see here is how variation in the thermal component maybe ranges from 600 to 900 across the collection of the material. And the photoperiod sensitivity ranges from 0 to something like 75, 85 units of thermal time per hour. But what's important is to look at here is the structure of that trait space, that trait variation. The tropical material is colored in green, the temperate material is colored in blue. And so you can clearly see that in tropical environment, for the tropical material, it tends to be late per se on average, and it has a lot of variation in photoperiod sensitivity. Whereas temperate adapted material is wide range of per se variation, but almost no or very limited residual photoperiod sensitivity. And what this has done, so what when we look at this, we realize that this is left like a hole in the trait space for maize. So all North American maize or European maize is basically selected strongly for moderate to early flowering time, and that flowering time having been funneled through the adaptation to long day length has lost its photoperiod sensitivity. But that doesn't mean that breeds couldn't be developed that fill this particular hole in the space, because that combination of very early per se and moderate or high degrees of photoperiod sensitivity could still lead to the same flowering in days in a northern environment. But what that would take is it would mean that you'd have to select for early flowering in a short day length environment long enough to hang on to that photoperiodic variation. If you remember what I showed you about the experimental evolution, when you select for earliness per se, the photoperiod sensitivity just drifts. So it stays, that component of variation just hangs around. So as long as if you were to select for things that were, say, in this zone of earliness per se, in a short day environment where you did not induce photoperiod sensitivity to be exposed to selection, you could fill things in this trait space. And then the question, of course, will that be advantageous in some way? Is it advantageous to have genotypes that now as they lead towards their flowering times, be controlled not only by temperature, but also by this combination of temperature and photogrid. So we're doing some work to try to address that question. So the last part I'm going to get into is model-based coupling of the ecophysiological model and genome simulation. And this is a bunch of parts to it. So I hope I'll just letting you know in advance, there's several pieces to the puzzle, and I'll try to do my best to communicate this well. But what we're essentially doing is we're trying to virtualize the genetics, plasticity, and evolution in one singular modeling framework. And what this graphic is showing here is how we use training populations to compute genomic predictors for ecophysiological traits. These traits that we're measuring are organ scale responses to environment. So like how fast do leaves emit as a function of temperature. This is a classic physiological trait called phylochron, but it's just how fast leaves emit as a function of temperature. So if we measure in a panel how fast leaves emit as a function of temperature, and we express those as sensitivities, we can make a genomic prediction model for that and embed the architecture of that genomic prediction model into a genome simulator. So we have kind of a realistic representation of the architecture and extant diversity of maize that we embed into this system. We embed recombination rates from real-world populations. So we have recombination maps that are determining how recombination occurs within maize, and we have real genotype data, so real parents that we're going to make crosses of. But we're going to end up doing that, combining all of that in a genome simulator that then virtually reproduces crosses and their traits with respect to ecophysiological variation. And those ecophysiological traits, like the rate of leaf emission as a function of temperature, is one of several parameters that feeds into a crop growth model, and that's depicted here by these complex interactions. And those traits interact with conditions of the environment. So then we plug in information about the environment. We basically are just using the crop model and putting it into the framework of population genetics, if you want to think about it in a simpler way. So the crop model then will reproduce the phenotypic outcomes as a function of the parameters of those virtual genotypes that were predicted via our genomic prediction model and formed as a consequence of the virtual recombination process in environments that we choose. These could be real environments. These could be environments that have hypotheses about climate change, as we wish, provided we have real, accurate models. And then that generates data that can be used for analysis of G by E and also starting selection cycles. So then we can take our virtual recombinants, apply a selection cycle, feed them back into the simulator, recombine them virtually, and go recycle this process. So one of the projects we have right now is we're questioning how will G by E evolve under warming. And so probably this graphic is not aligned with the way I presented at the beginning with what we should expect, but the main question for us is the extent to which we can ask how G by E, whether it be scale change or rank change, G by E will evolve over time as we think about our breeding strategies. So this is the kind of the multiple pieces. I'm just going to highlight a few things with a few slides to just demonstrate some of the aspects of it. But as I mentioned to you, number one is what we have a way of measuring some of these traits and then estimating those ecophysiological parameters, embedding the architectures of those parameters in the genome simulator, taking real parental genotype data and simulating them with real recombination data, and following some kind of pedigree structure. In our case, we tested this with some of the NAM population structure as initial validation scheme, using the virtual recombinants that carry forward, that inherit these ecophysiological traits that are genetically determined, and then those fit into a crop growth model where we combine it with environmental data from satellites, and then finally predict phenotypes of unobserved genotypes and unobserved environments. Okay, so this is what I just described, and that's kind of just a textual version of what I mentioned. So let's just- So what I want to share with you in a few slides here is just a few of the snippets of that and a conclusion or a finding from doing that work. So first of all, at the labs, we have a robotized phenotyping platform that can accommodate 2,400 plants. The plants are fed through that system on a every other day type basis, where computer vision scientists in the group and engineers in that group are able to extract phenotypes from 3D from RGB images that are then constructed into 3D models or virtual plants like you see here, where the latest things is they're starting to be able to track individual leaves. And then once you can track individual leaves, you can characterize specific features of the leaves. But in any case, we use the platform as a place where we characterize these ecophysiological traits. We measure them. And then by measuring them, we embed those architectures for what I'll just call here E-traits, like I said at the previous slides. And then this is what happens when we take our genomically predicted parameters. That will be, this parameter is an intercept that reflects basically the number of preformed leaves that are present in the plant and just before they emerge, and you start scoring the rate of leaf emission. So this is one parameter. Another parameter is the actual rate of leaf emission, how quickly leaves are formed as a function of temperature, and then the final leaf number. And it's easy to think about. You have one genotype that makes 20 leaves, another genotype that makes 25 leaves. So the rates of leaf emission can differ for those, and that can lead to outcomes of the same flowering or different flowering, depending, these combinations of characteristics are basically a part of the main drivers of flowering time. So you can see with our BLUP, the prediction accuracies from tenfold cross validations of the individual parameters. And then if we plug them into the formalism, which is this kind of function shown here as a graphic, not a real function, the cross validation accuracy for flowering time itself in thermal time is 68%, which compares pretty closely to what we would get if we were just to make a direct flowering time prediction accuracy. This is just showing us that the ecophysiological traits that we're trying to use as parameters for the modeling system are predicted with reasonable accuracy, and that we should expect this to be comparable to what we could do if we were to just focus on the output of flowering time itself. Secondly, the simulator we're using is called SLIM. It's more used in evolutionary simulation, but we chose it for reasons we could talk about another time. But essentially, what we've done is we've taken real genotypes, followed the kind of readings. These are rills that we're making, so they're just biparental crosses at the moment. But we took real genotypes, followed the rill formation, used the consensus recombination map for maize, and then simulated virtual recombinants. What this graph is just showing you is how the real progeny, which exist, so which have the same genotype markers scored, and our virtual progeny overlap perfectly in the PCA space of these particular crosses: B73 by Ohio 43, or B73 by B97, B73 by MS71, B73 by NC358. So we chose to work initially with these four populations, and you can see how they overlap. So that's just really a validation that the model of meiosis and recombination to form virtual genotypes is working pretty well. That was a check to make sure that we weren't deviating in that space. And then what we do is now we're going to finally connect that to the E-traits and then simulate them in the specific environments where those NAM populations were grown. And that this graph starts to show how the distributions, the phenotypic distributions of flowering time for the virtual and the real populations overlap with one another. So on this x-axis of variation, you have different environments. I think there's 8 environments here at the four different crosses on the y-axis here, and you're just looking at how the red and the blue kind of overlap with each other, and we end up with a mean bias in the timing of flowering of -1 to -4 days. And some populations, show a slightly higher bias than others, but there's this kind of strong, overlap in their distributions. So we move forward in trying to scale this out and model this then more broadly in population crosses that weren't constrained to NAM or the environments where NAM was tested. And immediately, one of the things we did is I think I just got a few more slides here. Looks like one of the immediate things we did was reorient ourselves in how we would measure flowering time variation. So I'm coming back again to the scale and rank change G by E aspect here. And when we were thinking about how to summarize the variance of flowering time, we ended up deciding to focus on the flowering window. And the reason why I bring this up is because if we think about a population that's planted at a given time, so what this graph is showing is time in days and average air temperature in blue, and the population kind of flowering this colored in this window. So if we think about we plant a population at a given time, ignoring any microenvironmental variation or ambient gradients of temperature, everybody in the population is experiencing the same temperature conditions all the way up until the stage of the first plant to flower. Once the first plant flowers, then all things change after that. In other words, the individuals that haven't flowered yet experience new conditions. It's a terminal thing. So the last individual to flower experiences a whole bunch of temperature scenario conditions within that window that the first plant to flower never did. And consequently, if you focus on the window, this is a way that we can summarize the variance in flowering time. I'm not talking about the mean anymore, I'm talking about the variance. And you can see here in one environment where we might have a cold snap during the flowering window, you can get a real expansion of the variability compared to an environment where you have a warm spell that happens during the flowering window, where you have a compression in days, okay? That leads to that scale change G by E that we were talking about at the beginning of the talk. So we simulated this out in 2066 environments across the US and Europe. And in this case, it's from data that we had the real planting dates and environmental conditions of where these maize were for these particular maize fields. And we made 2,000 virtual rills by crossing 10 populations, by making 10 crosses and 200 rills for each population, and then grew those through this process across all these populations. And what you can see from this graph here with now we look at the variance in the flowering window against the mean temperature in that window. That's what's plotted here. So you have the mean temperature across the flowering window on the x-axis and the additive variance on the y-axis. And what you can see with this blue, line, we'll focus on the blue line here. There's a reduction in the phenotypic or the additive variance by about 50% with every 3 degrees C. So that's kind of the pattern of fall and the way that the variability shifts as we go from roughly a mean of 15 C to about 30 C in the flowering window. So that there's like this kind of average response. And then separate from that, of course, you have lots of deviations around any individual line. And that's basically a result of the cold and hot wave. So you can have within the flowering window the same mean temperature, but yet in one place you get a cold wave, in another place you get a hot wave. And so consequently, that leads to these vertical deviations around the variability. If we rescale that to a heritability, making an assumption about the residual, it linearizes, like in the graph shown here. And in that case, what we see is a range in heritabilities of about 90% to about 60%. And we have a total of about roughly 35–40% point range in heritability across the temperate zone, because the modeling we did was just across the temperate zone, and an average of a 3-point reduction in heritability per 1 degree C in a given environment. So if we were to, so the kind of take-home message here, at least the kind of preliminary point of view in questioning the impact of climate change on breeding, is how can we think about what factors of the climate will alter the variability in the traits we're trying to select. In this case, what I'm showing you is how warming causes a compression and variance of the flowering in day. We measure it in days, giving us less discernibility of genetic differences, and we should expect that to lead to a reduced progress in our selection response. We tried many things to see if we could compensate for that, like shifting the planting dates and other manipulations of the the response equation. And the main thing that it looked like we'd have to do is ratchet up the selection intensity. So that has other consequences. So what I shared with you today was how interlinking ecophysiology and quantitative genetics enables modeling of this genetic plasticity and evolution. That's kind of the glue that's allowed us to do the work I presented today, the GP meta process underlying adaptation and how we're thinking about adaptation as this process. And that this helps provide kind of a mechanistic view or hypothesis. And so it takes us differently from a statistical space to a mechanistic space of thinking about the formation of G by E. And one thing that's interesting about this, I think, is it exposed the importance of intra-environment G by E, which is something that we often neglect or haven't had the capability yet, and still may not, of capturing that or knowing how to model this. But as you can imagine, that very likely creates a lot of confounding of the types of modeling we do when we do cross-environment G by E analysis. Scale and rank change G by E can be explained by genetic variation in the environmental sensitivities at different timescales of development, and that this leads to these context-specific effects of adaptation that have shaped plasticity of modern breeding pools. I mentioned to you about the hole in the space and how it helped expose this scenario that may open up a new channel for breeding to be carried forward and questioned. And that the coupling these process models that include, in our case, processes of genetic transmission with crop growth within its lifespan enables a meta-modeling approach for larger scale virtualization, so we can do things that we can't just do in our real breeding programs, and it just opens up an exploration space to test different questions we might have. And the implications are not only for crop breeding and what I shared today in terms of those. I didn't get into this, but in the back of Lynch and Walsh's book, they've got a formulation of how to derive the expected variance for a given response function, mean response function. And that blue line I was showing you as a relationship between mean temperature of the flowering window and the additive variance is actually derived by fitting that mathematical equation to the response function known for maize temperature response. And so you can so I say that to say that there's a much broader implication whereby because temperature is so fundamental to all species and their development, that even natural populations should expect their variance scale change to be affected. And meta-modeling basically is a framework that we're finding is an opportunity for disciplinary convergence. We're working with ecophysiological modelers, computer scientists, computer vision people, and quantitative genetics. Combining these models is a way for us to have discussion, dialogue, and connect with each other. And so that's the direction we think is real promising. With that, thank you. There's many people to thank. I mentioned several people who have contributed to the work here, but there's several others that have been present throughout a lot of lectures. Thank you.
Thanks a lot. Anybody questions?
Yeah.
So that I was interested in intra-environment that we even talked about, of course, flowering had that standard of the planet.
Yeah.
So do you think that this could explain maybe some lack of reproducibility between so not accounting for that? That's actually very different environments.
Yeah, I think it's like a confounding source. So there was you could do this thought experiment where you say, okay, because we thought about things like this. If I think there's just a component of this that will never separate. It's inseparable. If you think at a population way, you could do that thought where you say, I'm a human being, my brother, my parents grew up in a different environment than I grew up in, doing plants too, right? What if I was born on the same day of my parents, grew up in the same environment with the same grandparents as my parents? Would I be just like my parents? Whatever. Or you can think about a population. We can actually model this. We can use this virtual system, grow a population for flowering, select individuals for earliness, and recycle them back and grow them right back in the same exact environments where their parents were grown. And as soon as you go through these thought experiments, you already know that there's going to be a component there that is completely inseparable. The genetics determine for you, what is the environment you experience. It cannot be the same. So there's a part of this intra-environment stuff that's completely inseparable. If you think statistically, you could say you have the axis of genetic variation and axis of environmental variation is needed to have G by E in the first place. But the axis we're talking about is the temporal axis of environmental variation, not the locational axis of environmental variation. So the source of variation here becomes temporal for environment, and for genetic, it's just the individuals. But that leads to this intra-environment component. The question is like, how much of that can we separate? But I think there's a part of it that's just not separable. But the part that we can separate, the challenge would be, how do we measure the individual environment condition that each genotype in the same place is experiencing? So I mentioned about photoperiod. We have a way to say, within this environment, this genotype experienced 16 hours, this other one experienced 17 hours. So now if I want to model the G by E, I use that as regressors, for a cross environment analysis. And I can pull out a little more of that, what was probably confounded variation before is moving to the residual, because you were treating that environment like everybody had the same mean photoperiod in the past. All the individuals here, their photoperiod, whether it be a window of time or not, you've just assumed that's fixed for everybody, and you do cross environment G by E analysis, and you see where is that clear? Whereas now we should be thinking like, how can I measure the environmental conditions that different individuals are experiencing within the same place to understand the structure of that G by E?
So in terms of the growth aspect, so you use the temperature, so temperature and photoperiod.
Yeah.
And when you think about growth, when it comes to plants, it's a function of temperature,
Yeah.
cell cycle, etc. But there's also the expansion part, right? And the expansion part is in large part driven by hydraulics. There's several rheology, etc., that control temperature through cyclic processes. So if hydraulics are involved, mean that evaporative demand will play a major role in filicrine also.
Yes.
So as you look at temperature and photoperiod as main environmental drivers, how do you integrate the role of
Same way, actually. So that's like another edge of that's the trailing edge of some of the stuff we're doing. So in the platform system, where we've used our well water treatments to parameterize some of the things I talked about today, we have done water deficit work to try to get the sensitivities to water deficit for several of these traits. And then they just in the end, they all just become different levels of sensitivities, how far we can go with that. So we're looking at, we started with temperature and photoperiod for several reasons of availability of information, of more available, even in the fields where we could try to validate against fields, getting proper water deficit information about the field conditions experienced by whatever we're measuring can be a bit difficult to. Anyway, so we're focused on temperature and photoperiod, major drivers. Water deficit is the next target alongside light intensity, because so there's a notion that you can get you're trying to get something like when you talk about thermal time, it's something that we could say is an intrinsic trait. It's like we've kind of removed the effect of temperatures impact, and we're getting down to something kind of intrinsic about the genetics. But of course, when we reduce to phylochron, temperature response, and we test it in different light intensity, the phylochron rate itself, which is a function of temperature, is also a function of light intensity. And so then so now you can add the light intensity and re-express the sensitivity as a function of both temperature and light intensity. And then you think, okay, now I'm on the intrinsic thing, right? But then, okay, wait a second, there's water deficit. And it's another one. So how far you can get down and reduce to get to something that's really intrinsic is an open question. But we're certainly see it that we can at least get the major components of this model and then use it in a way to establish sort of maybe a more direct way to go forward, or test different ways to go forward and how it's valuable than necessarily being exact in our.
I guess what I'm implying is, part of what I'm implying is that, so an operative demand can have and will have an impact on leaf expression in complete absence of soil moisture deficit.
Yeah.
So what I'm saying is basically perhaps the temperature is also conflated with the effect of PPD.
Sure.
Yeah, that's kind of like, if you want to parse that out, and that might help explain some inter-environmental differences.
Yeah.
Because that like changes in urine diurnal inspection.
Yes.
I think your point about how the intrinsic aspect.
Yeah. So PPD would just be another one. So yeah.
There's a question online. It says, can we single out the effect of one environment covariate effect like temperature in genome prediction rather than the aggregate effect of others?
Can we single out the
Effect of one environmental covariate.
covariate?
Yeah,
Yeah,
like temperature.
maybe not in natural environments. I mean, I think we have a problem in quantitative genetics that we just don't. I mean, not at least maybe in public research. Maybe for some of the companies that started, where their number of environments they can test is extremely large. Our biggest problem is we have none of us would accept sample size of less than 20. We're talking about, quantitative genetic inference or whatever. Even 100 is already quite limited for us. And we know that there's a lot of covariance in that. So the answer to that, I think, is related to our limitations of, our understanding and limitations of environmental covariance and making meaningful interpretations about environmental effects. And that also depends on the scale, the environmental scale at which you operate. So we've done work where we've operated across broad latitude, and when you go across broad latitude, you have a lot of, covariate effects of the environment. So this is probably the biggest issue of trying to dissect environmental components from our field network trials.
So the idea of temperate early flowering photo periods that would make sense. You said thinking about that. Yeah. When underneath cycle, would that be advantageous for you to determine? Not just
might not be advantageous. The things that happen, things we've seen happen not with early flowering. I mean, the thing is a photoperiodic effect, and I don't know how much it's fully coupled, but one of the things that happens with when we select early photoperiod is we get reduced number of we increase the number of cupules that form on the ear. So basically there's some kind of coupling between the photoperiod sensitivity and the yield and grain number, and we don't really understand right now. That's all related to the timing of the floral transition and leaf formation and development, but we don't understand why that is. Maybe it's a sink source effects. We're not sure. But when we've taken in the parallel selection experiment, we measured, kernel number directly as cupules numbers, and from that you can see as populations become more earlier as a consequence of reduced photoperiod sensitivity, we see a corollary impact on the grain number. So I don't know, so whether that's purely due to a photoperiodic impact, or whether that's separable, I don't know right now, so.
That's a longer period of development that hit by more.
The question there is whether this also happens, I just don't have material like that in that space, or we have a few things in that space now that we're testing, but they're rare, and so we just need to see, if it's just due to the pure duration of the cycle, or if it's actually connected directly to photoperiod sensitivities. Yeah, because it would be, if you're photoperiod sensitive, if under short day, do you even see this type of an impact?
Okay, we've reached the time. So thank you, Randall.
Okay, thank you
for
very much.
this